So again, today's lecture is all old stuff, with the exception of the first, I guess, 5 slides, one of which is this. The next 3 are clickers, and then there's the outline. And again, everything else is going to be reviewed. They're exactly the same PowerPoint slides that I've used before. 2 reasons that I choose them. 1, I think that I did a really bad job explaining them the first time through. And secondly, these are things that I think are particularly important. Doesn't mean that they're going to be on the exam, but things I think are particularly important. So this, of course, now turns out to be the one I think that doesn't have the clicker questions. So you miss out today. Sorry. I could reload them, but should I? No. Yes, no. Yes. So I should give 3 points to everybody? Yes. Yes? Even the people who forgot their clickers today? Yes? So I will post them, actually. That's a good point. I will post the clicker questions. I had a problem with my bike this morning. Actually, you're lucky I'm here. So that's why they didn't get transferred properly. So I will post those clicker questions with the answers to them. And if people have questions about why those are the correct answers, send me an email, and I'll try and go ahead and explain them. And those of you who need to take off, and you were just here to collect clicker questions, leave, and please leave quietly. So what we're going to be talking about today is really just the stuff which is going to be on the midterm. So that started out with DNA damage and repair, particularly homologous recombination. We spent quite a long time talking about homologous recombination. A little bit about viruses, nowhere near enough, which is a quick reminder. If you want to take virology next term and you haven't taken cell biology yet, that's okay. Just either talk to Leah Tour in the biology office or talk to me, and we can approve that. If you haven't taken cell biology, however, there will be some things in virology that will be a little bit challenging, and so I can help you out with that. Classical and molecular genetics. This is the lecture which I posted online because we were canceled that day, and so I'm going to spend a little more time talking about that. Human genetics, gene disruption, and then basically the last parts of chromosomes. I like to think of proofreading, which happens via DNA polymerases, as sort of the first line of DNA repair, because really you're putting in the wrong base pair. And then if you put in the wrong base, I should say, wrong base in a base pair, that's where you first need to have some kind of correction materials or some mechanism for dealing with that. And the way that all the replicative polymerases do that is they have a separate domain which has a 3' to 5' exonuclease activity, which is backing up. So you're always going to be polymerizing from 5' to 3'. If you back up, that's going to be doing a proofreading reaction. So all of the replicative polymerases have these. Some of the error-prone polymerases don't have this particular domain, and you remember, hopefully, or you've been studying, that we've got some of these error-prone polymerases that will put in just whatever nucleotide. Those don't have this separate exonuclease domain for dealing with these particular mismatches. So if you put in a base, it could be the right base, it's still highly susceptible to DNA damage because we're at 37 degrees. Some organisms, of course, hotter than that. We live in an aqueous environment, and we're exposed to light, I should say, life too. So the main things which are happening here are deaminations of cytosines and depurination. So where these big blue arrows are, and that's due to water activity, which is cleaving these guys off or modifying them in some way. So here, cleaving the amide group or cleaving off the purines. There are 2 major ways for dealing with this kind of DNA damage. One is the really specific way, so all these common kinds of changes that you have, the deamination of cytosine or depurination. Very often you have a specific enzyme that will recognize that problem, in this case, a deaminated cytosine, and then that will be very specifically repaired. Just one single base will be chopped out and put back in. If, on the other hand, you have a more rare kind of change which happens, or something which is a big bulky change and not something which that single base repair can deal with, then you chop out a lot more and that's this nucleotide excision repair. And this is a much less specific mechanism and much less efficient because you have to then refill a whole bunch of your DNA. But since it's a nonspecific mechanism, you're looking at the structure of the DNA as opposed to that actual specific base pair change. And so this is really dependent on looking at structure, which is exactly how you get mismatch repairs to be taken care of. Because again, if you have a mismatch, the structure of the DNA is going to be incorrect, but both of the base pairs on each strand are actually correct bases. So you can't have a specific machinery that says, okay, we want to take these out. So in the case of E. coli, mismatch repair, this is through the action of proteins that are known as the mutation or the MUT proteins. That'll be important in just a second here. Bind to, again, this aberrant structure that you have in the DNA, and then you have cleavage on either side of this and repair, which takes place. Human mismatch repair actually functions in an extremely similar way, and the only reason that I wanted to bring this up is that the naming of these are the MUT-S homologs. So these are similar proteins to MUT-S and other proteins, MUT-H in particular, that you have in E. coli. But it's just, naming-wise, to try and deal with some of the alphabet soup that unfortunately you're exposed to in this class. That's what this means. So it's a MUT-S homolog. MUT-S, is important for E. coli mismatch repair. That way you should know what MUT-S or MSH is important for. But again, the mechanism here is basically identical. Oh, by the way, hold up your hands and stop me when going through some of this stuff too fast. Double-stranded breaks, these are particularly problematic because it's not a change in the structure of the DNA. It's not a change in terms of one particular base. And once two ends come apart, they could come back in all kinds of other different places. And I don't know how many people watched Brian Drucker's talk, UCLA, the other day, but that's exactly how the Philadelphia chromosome forms the BCR-ABL protein, which leads to chronic myelogenous leukemia. So it's this problem with double-stranded breaks getting put back in the wrong place, which can lead to some really serious disease. Two ways of dealing with this. There's the fast and dirty way, which is chop off the ends a little bit so they work with each other and bang them back together. That's the non-homologous end joining. And that, of course, leads to degradation of whatever sequence is between, actually right around where you had that double-stranded break. The same thing actually happens in homologous recombination. Whenever you have a double-stranded break, you cleave in at the ends, but you use your homologous chromosome for the information to repair that. And so you don't lose overall nucleotides here, but you could be changing the sequence of the nucleotides here if whatever this homologous chromosome is that you're using has a different sequence there. And so this is the process also known as gene conversion that can be taking place. So you've taken whatever gene, or better spoken in the genetic terminology, the allele, which you happen to have on the red chromosome, and now you've replaced it with the allele that you have down here on the orange chromosome. You can see that a little better when we look at the overview of how this happens. We mentioned that double-stranded break repair is important for dealing with double-stranded breaks, but you also have to have double-stranded breaks in order to have meiotic recombination. So probably why sexual reproduction was developed in the first place, developed, i.e. evolved in the first place, is using this mechanism. So we have this specific nuclease complex, particularly here, this MRE, by the way, stands for meiotic recombination. So you have a mutation in the MRE complex, then you have a defect in meiotic recombination. So this will cleave one of two pairs of your sister chromatids, chew in at the ends. You now have a free 3' end, and this is true for all homologous recombination, not just meiotic recombination. Once you have a free 3' end, Rec A binds to this in bacteria, Rad51 in eukaryotic systems, and that stimulates this strand exchange where you have this free 3' end which will now go in base pair with its opposite strand. It has to have the presence of one of these recombinase proteins. So Rec-A, Rad51 also depends on having ATPase. And so this just doesn't happen if you have a single strand floating around in a double strand. You have to have these strand exchange proteins. Those are actually absolutely critical for getting this reaction to take place. Once you have strand invasion that's taken place, this is great because now you have a 3' OH that you had up here. Now it's got a template right next to it. The cellular DNA polymerase can extend this with these green pieces right here. And then you have ligation that takes place between this extending DNA polymerase and this 5' phosphate, which you have here. This process and that ligation generates holiday junctions. And whenever you have a double-stranded break repair, you're always going to have two 3' ends, and each of those is going to invade another double strand in some way. So you're always going to end up with two holiday junctions whenever you have double-stranded break repair. So this is one holiday junction, the second holiday junction. This is drawn here as just an X and two straight lines. That's really not the case. It's much more of a cross structure, and that's because the double helices are wound around each other. And because of that, these structures are completely symmetrical. And since they're symmetrical, you can cut them either across or up and down and religate them. And so the machinery, at least in theory, doesn't know which way it's going to cut. Are you going to cut in such a way that you end up with chromosomes that have the same information on either side of these two holiday junctions, or those that have different information on either side? And when I say same information, I'm talking about this flanking chromosomal sequence, which is on the outside here. So here, in the case of the two holiday junctions cutting basically in the same way, this can either be, in this case, horizontal or vertical. As long as both of them are the same, you're going to end up with the so-called non-crossover. So both ends of your sister chromatids are going to be the same afterwards. If, however, you have them that cut orthogonally to each other, one vertical, one horizontal, doesn't matter which one, then you're going to end up with one of these crossover events, where now we've got flanking chromosomes which actually came from different sister chromatids that they started with. Okay, people clear, unclear, confused, overwhelmed because it's midterm season and we just can't think anymore? Okay, so that's it for DNA repair. Wanted to really briefly cover some more of virus stuff. Actually should have brought a plate today because I got some plaques this morning from my overnight incubation. But since we didn't have a chance to talk about lambda bacteriophage when we had our virus lecture, I wanted to talk a little bit more about the lysogenic lifestyle, because that's something which is probably really common in probably a majority of viruses which are out there on our planet, and there are a whole bunch of them. But also because the process of integration has actually taught us a lot about things like recombination and transposition, etc. So these particularly, lytic is pretty clear. Virus infects a cell. Cell makes a whole bunch more virus. The cell breaks and all that virus is released, goes on and infects another cell. Chronic infection, virus gets in, makes a little bit, convinces the cell to make a little bit more virus, and then that cell actually persists for quite a long period of time, and virus is produced for a long period of time. Lysogenic actually is really kind of nice because it's kind of in between these two. What happens is the virus infects the cell, gets its genome on the inside, and then basically it makes up its mind whether it wants to be lytic or hang out and wait for different conditions. And usually those different conditions are the cell is reproducing either really rapidly, so you can produce lots of virus, or there's some kind of DNA damage. And in fact, the SOS response was originally found because the virus was being induced in that particular case, because there was DNA damage and that virus had sensed that there was DNA damage. So what happens is Most cases, you have an integration of that virus genome into the host genome, so a lot like what's happening with retroviruses. Only once that genome is integrated, it's actually not functional. It has to be then chopped back out of the host genome in order to be replicated. And then you undergo virus lysis. This HIV and retrovirus infections are usually much more of these chronic infections, and they'll just be continually producing virus. In some cases, the cells that they're infected will die, but in many cases, they're just producing small amounts. So lysogeny, so the genome integrates, what's called a prophage, and then these prophage can be induced, and that leads to the lytic cycle of replication. And again, bacteriophage lambda is the important one for this, and take virology, and we'll talk a lot more about it and we'll move along. What I didn't bring this morning were my plaques. Again, Petri plates with some spots on them where the bacteria did not grow, and that's because of the activity of a virus, and in this particular case, a lytic virus, because it has stopped the growth of all of the cells that were around the one virus-infected cell that you started with. And so this is a way of literally counting the number of infectious virus particles that you have in your solution. Really important here is that this is a dilute suspension. I diluted my virus preparation 1 million times before I put it onto the plate, and I still saw about 100 of these particular plaques that were forming there. So you get lots and lots and lots of virus. And then incubation, and these plaques, again, are due to death of first that original cell that was infected, but since you're on a plate, there are lots of cells around that which can also be infected, which produce virus and infect the cells around those. So each of these individual plaques actually represents millions of viruses that have replicated through a number of cycles by the time you take that plate back out. Seems to be some confusion about terminal redundancy and circular permutation. This we talked about in the context of bacteriophage T4, but it's a pretty general concept, so I wanted to talk about it a little bit more. All that terminal redundancy means is you've got the same information at both ends of your genome. So here, in this particular genome, A and B are terminally redundant. Now, it doesn't matter if this is a circular permutation or anything. So this particular genome, the top one on here, is terminally redundant because it's got A and B. The second genome is terminally redundant because it's got B and C at both ends. The third one, C and D. So these are 3 different terminally redundant genomes. And it's just, again, it's the same information at each end of your genome. Now, if it so happens that, this case where you've got, ABCD and BCD, and in the same case, ABC, ABCD at the other end, this is an example of circular permutation, because if you think about a circular genome that would start at A and go all the way around to A again, that would give you AB, ABCD. And so if you cut here, that will give you one genome. But if you cut here, it gives you a different genome. You cut here, it gives you a different genome. And now all that's saying is you have a linear genome that's basically like cutting a circle in a different place. And that's where you get your circular permutation. And so circular permutation is multiple different genomes, each of which is terminally redundant in the case of bacteriophage T4. It doesn't have to be terminally redundant. That, you can have circular permutation in the absence of terminal redundancy. But that circular permutation means you're linear, but you've started from a circle, and that circle's been cut in a different place. And any of you have been up to the 4th floor of the SRTC building, I like to think of it as sort of being terminally redundant if you, not terminally redundant, I mean circularly permuted. So if you start at the elevator and walk your way around, that's going to give you one thing. If you start at one of the stairwells and walk your way around, it's going to give you something else. So it's basically, it's a circularly permuted floor, because you can just, depending on where you start, you're going to still get the same thing, but the start and the stop is going to be in a slightly different place. That make more sense? Less sense? More confused, less confused? Who cares? Let's get done. Okay, so this is the lecture which I posted online. Hopefully everyone's had a chance to take a look at it. Basically, this is classical genetics. And I think I overheard that people just had a genetics midterm, or they're just about to have a genetics midterm. True? You just did? Yeah. So hopefully all this stuff is easy because you've just done it already, right? So basically, as far as this course is concerned, it's really about the identification of genes and how to study individual genes, particularly at the molecular level. So really two kinds of genetics, both so-called forward genetics, which is probably mostly what you've been talking about in the genetics course. Look at the syllabus, probably mostly the case. And then much more what's happening in molecular biology, and particularly these days is so-called reverse genetics. And so with forward genetics, you have a mutant phenotype, and you try and figure out what that gene is. So that's this downward leading arrow here. On the other hand, particularly in this day of genomics, you have a gene, and you try and figure out what it's doing. And so that's the case of reverse genetics, where you now start with usually that gene product, and then make a mutation in it and see what happens. And so there are two very different ways of actually really figuring out the same thing. Starting out with a phenotype and figuring out what that gene is, or starting out with a gene and trying to figure out what the phenotype is. So just flip sides of looking at exactly the same thing. Now, one of the really critical aspects of classic genetics, at least I like to think of this as classic genetics, and it's really made a huge contribution to understanding the molecular basis of a lot of how these things function, is the idea of a conditional lethal mutant. And usually these are temperature-sensitive mutants, but that condition could be lots of different things. So the conditional lethal mutant is you have normal cells, and in this case it's yeast, but it doesn't really matter. It could be bacteria, it could be human cells. You have either some kind of mutagenesis that takes place, or you're just looking at randomly screening for mutants. And you get lots of different possibilities that you now put at your permissive condition. And again, in this condition, it's a temperature sensitive. So you put these at a permissive condition. And then the critical aspect here is that you now need to screen for all of these different potential mutants. So that screen, the way you do that is you need to separate all of these different cells, hopefully one or a few of which are actually now mutant in the phenotype that you're looking for. And then you have to keep these guys at their permissive temperature and then test them at your non-permissive temperature. So this is really nice because this particular colony here, which has come hopefully from one particular cell, should be clonal, so it's just one particular mutation that you have there. This cell can grow at your permissive temperature, but can't grow at your non-permissive temperature. And so that says, okay, this gene product, whatever that mutation is important for growth under those particular conditions. And now you can go back and isolate the gene from this guy and actually figure out what it's doing, and then figure out what the difference is between this gene being mutated and one which is then no longer mutated, or one that's before it's become mutated. So this was the way that all of the CDC mutants were found, the cell division cycle mutants in yeast. And these individual mutants, of course, you don't know when you do that mutagenesis, and usually it's actually chemical mutagenesis, so you make a whole bunch of DNA damage, and too much DNA damage for it to be properly repaired. And you have a whole bunch of mutants, and what you care about is how many different genes you happen to have each of these individual mutations in. And this leads to one of the ways to do that, and probably the best way to do that, certainly from a genetic point of view, and that's so-called complementation analysis. And basically what you're looking for here is that one of your mutants, which is, again, usually in this case, if you've got a really critical gene for cell function, it's going to have to be a conditional mutant, that can only function under permissive conditions, but you put the two mutations together, if they happen to be in the same gene, that gene is never going to give you function. And so you're never going to get growth under your non-permissive conditions. Whereas if you have mutations that are in different genes, but are still giving you the same phenotype, and so you think about cell division cycle mutants, they're not going through the cell cycle. Well, that could be because of a whole bunch of different genes that are causing that problem, because the cell cycle is a pretty big thing. Lots of genes are involved in it. So you have two mutants, and this is your standard forward genetics, that both are blocking the cell cycle. You do a mating, put both of them together. Now you find that these cells can grow under non-permissive conditions. That means that each of your individual mutants that you started with are in different complementation groups, because that's what people first called them. And then we now know that those end up being in completely different genes. And if you get the opposite case, you put the two of them together, you still don't get function at your non-permissive temperature. That's an indication that these two mutations are in the same complementation group and in the same gene. So is that clear? Again, everyone else studied all this for the genetics midterm, so you're all in good shape. Okay, so moving beyond the classical genetics, the forward genetics, is much more thinking about reverse genetics. Well, there are a couple of real big breakthroughs here. Restriction endonucleases and PCR, which really sort of allowed this process to take place. Restriction endonucleases, these are endonucleases. They cut in the middle of a piece of DNA, so that makes sense. But the really amazing thing about these restriction endonucleases is they cut at very specific sequences. So they're specific DNA-binding proteins and specific endonucleases that will cut at particular positions. Now, why or how, maybe is a better way of thinking about it, did this actually occur in the evolution of life? Why do you want to have something that chops up DNA inside your cell and your DNA in your own genome? Well, that's because there are these nasty other genomes which try and get inside your cell and make more of themselves. And so those are the viral genomes. And so this restriction endonuclease is a viral defense mechanism, particularly against DNA viruses infecting cells. And if you remember bacteriophage T4, that has hydroxymethylcytosine in its DNA. Well, none of the cells have restriction endonucleases that cut hydroxymethylcytosine. So that bacteriophage T4 is now resistant to any of these restriction endonucleases. So, but particular restriction endonucleases, and it turns out there are lots of different viruses that have lots of different sequences in them, so many different restriction endonucleases that cut many different sequences. Most of these restriction endonucleases cut offset to each other, and actually that offset cutting is very similar to what you see in transposases. They cut at sequences that are not directly opposite each other. And because of that cutting at sequences that are not directly opposite each other, these now give you ends which we call sticky ends because they're now compatible to a potential different piece of DNA which has been cut with the same restriction endonuclease, or even a restriction endonuclease that just gives you this same sequence here at the end. So this allows you to chop up DNA from bacteria, from human cells. As long as it's the same restriction enzyme, those two can actually go together. How do you usually end up doing this? You use cloning vectors. These cloning vectors have origins of replication. This origin of replication could be an origin of replication that functions in bacteria, if you want to make this DNA in bacteria. Could also be an origin of replication that functions in yeast or in human cells. It's just, depending on where you want to be making more of that DNA, you'll have that particular origin of replication. Usually it's E. coli because E. coli is really easy to work with. You need to have some way of knowing that you've put this DNA into the cell, and usually it's some kind of antibiotic resistance, which you use for that. And then you have to have a place to put in your favorite extra piece of DNA. And so I mentioned polylinker in the last lecture. So this is an example of a polylinker, just where you have multiple DNA sequences that lots of different restriction endonucleases can bind and cut. And so it may turn out that your favorite gene from your favorite organism doesn't have a site for ECHO-R1. It doesn't have GAA-TTC in its genome, so you can't use that restriction endonuclease. But hopefully it's got BAMH1, GGCAT if I remember correctly, in your sequence. And so you can just pick and choose which one of these you want to use. So you do that. You've got this piece of DNA. You put it into your plasmid, small vector. And then now what you want to do is figure out, A, which one of the cells you have the particular piece of DNA that you're looking for. And so there are a number of different ways of doing that, but probably the most straightforward is using what's called a library. And this library is having lots of these individual E. coli colonies here. And each of these E. coli colonies is resistant to the antibiotic because it has that vector in it with that antibiotic resistance gene in it. And each of those colonies will represent the offspring, if you want to call it that, which then has replicated from that single cell that you started with. And so that will have one particular DNA in it and usually one particular plasmid in it. So one of those will have the gene that you're looking for. And anyone who's worked in a lab and tried to do this, lots of those colonies will have something else in them. But if you're talking about trying to screen for a single gene, say, in the human genome, if you've got 20,000 colonies, because we've got about 20,000 different genes, you're going to try and find that one colony that has the gene that you're particularly looking for in it. So the most common way to do that is to make these individual cells. You can't get 20,000 colonies on plates, although you can get really big plates that you can get a couple thousand colonies on. And then basically make a copy of those by pulling a small amount of those cells onto a filter, and then taking this filter and hybridizing it to some kind of probe which specifically recognizes that DNA that you're looking for. And usually it's a very short piece that you're using as a probe, and this is very often actually something which is synthesized. And you go to a particular website, you type in the sequence that you want, and lo and behold, the next day with FedEx it shows up in your lab. So that's really the process that's very often used for these labeling experiments. You know, a short piece of a particular gene, you want to find the whole gene, you use one of these probes. That probe now will identify one of these, again, we're talking about the human genome, one of the 20,000 individual colonies that you have there, and then you can purify the vector from that particular colony, sequence the DNA that's in that vector. It'll have this labeled DNA sequence plus the whole rest of that sequence. And so you can use that now to get the copy of your gene, that needle in the haystack that you're trying to find there. If you know a little bit more about that particular sequence, and in this day of genomics, you pull up a website and say, okay, I think this is the gene I'm interested in. How can I see if I've got that particular gene in my sample? Then we use the polymerase chain reaction. And I think I didn't do a terribly good job of explaining this last time, so I wanted to explain it a little bit more. There are a couple of things that need to happen in order to get the polymerase chain reaction to work. One is you have to have these known sequences that you order DNA sequences that will bind to. And so these are now DNA primers that you can use for DNA replication. So they've got 3' OHs on them. They're completely normal DNA. These now can base pair to the ends of the fragment that you're trying to amplify. You denature by just heating these two strands apart, or heating up your solution, the two strands come apart. Then you slowly cool them down, hopefully such that your artificial primers will bind here before the two strands come back together. And then, together with a DNA polymerase, these will now extend, and they actually will extend until the DNA polymerase falls off. So they're not just going to extend on one strand to this position. This DNA polymerase has no idea where that other primer is. So the first couple of rounds of amplification that you do with PCR, you're actually generating pieces which are far too long. It's only after you do denaturation, renaturation with your primers that you end up amplifying pieces which have the correct length here. Now, another aspect of PCR, which you actually don't need every time you do this cycling of heating up, cooling down, extending, etc., you can add DNA polymerase every time. That's actually the first way the PCR was done. It's a pain in the neck to add polymerase every single time. So if you happen to have a DNA polymerase that can survive high temperatures that you need to get the two strands apart, that will really, and it actually did, revolutionize PCR. Because now you can just put a DNA polymerase in at the beginning of your reaction. It will extend every single one of those templates, all the rounds that you put it through. And so it's the study of hot springs, like I do, and the organisms that were found there that led to these kinds of polymerases and the development of PCR. So any questions on that before we move on into DNA sequencing? I mentioned at the beginning of lecture today, Illumina has a brand new sequencer on the market. They're trying to get us to spend the money to buy one. But basically, Illumina sequencing and, for that matter, the old school Sanger sequencing really depend on some very similar techniques, and that has to do with the addition of fluorescent nucleotides and detection of the addition of some of these nucleotides through fluorescence. The classical Sanger sequencing or dideoxy sequencing is called chain termination sequencing, and so you have a mixture of nucleotides. Some of those are dideoxy nucleotides. So you end up with a population of DNAs that are all stopped at different positions, so a different length piece, stopped again by the addition of a dideoxy nucleotide. As long as you do 4 reactions and have the right concentrations of all of these things, you'll end up with a whole population of DNA molecules, each of which is one nucleotide different from the other ones, and each of which will be labeled with one specific fluorescent nucleotide at the end. So you can separate these either on polyacrylamide gel electrophoresis, or the modern sequencers actually do this on really fine capillaries, also filled with proprietary material, which is basically polyacrylamide. And all that they do is they have a laser and a detector of fluorescence at the bottom of that capillary tube, and they just run those DNAs off the bottom. So remember, you've got a population of different length DNAs, each of which is going to have a different fluorescent nucleotide at the end. And so these colors here represent, okay, the first nucleotide that comes off the bottom here has a T at the end of it. This nucleotide that comes off the bottom has a G at the end. This one has a G, this one has a G. This next one has a C, this one has a G, this one has an A. This one has a T, this one has a T, etc. And so just literally reading off from the shortest to the longest, each of which has these ends. And you can see here that this was actually not a really good sequencing reaction, because you end up with a lot more G at this position, a lot less at that one. If all of these peaks are flat across the whole sequencing run, it's actually a lot better. So this is not the best example of a good sequencing run. I talk a little bit about pyrosequencing. Basically, again, this is not in the text. There's actually a lot of really good information online, so if you're interested in pyrosequencing, you can take a look at that. But the important thing here is that we're not looking at chain termination. We're looking at the production of pyrophosphate, which is part of that extension reaction. So when a DNA polymerase adds a nucleotide, it generates pyrophosphate. And if you can detect that pyrophosphate for one given nucleotide, then you can use this to show that you're actually getting polymerization to take place. And this is nice because you can do it in a really high throughput format. Illumina, guys are trying to sell this new sequencing instrument, basically does a really, oh, sorry, question? Yeah. Yeah, so the way to get this specific to one nucleotide is literally what you have to do in your pyrosequencing reaction is you have to have all of your parallel sequences, usually hundreds of thousands of them, and you wash them all with one particular nucleotide. So ATP, for instance, or DATP, I should say. And then you look to see where A is being added. You wash off all the A, and then you add G. You see where that's being added. You wash off all the G's. You add T's. You see all those are being added, etc. And so you literally have to go through one nucleotide at a time and do that, which actually is not that dissimilar from what happens with Illumina sequencing, because you're also now putting in the individually labeled nucleotide triphosphates, but now instead of detecting the pyrophosphate, you're actually detecting the addition of the nucleotides. And the addition of the nucleotides here, they're fluorescent nucleotides, but they're not chain-terminating nucleotides. They're nucleotides that just keep getting added to the primer template which you have, which you've generated through PCR on this surface. And then you can see through the different colors that get added in any given cycle. And you just put in enough for one nucleotide to get added. You look at that one nucleotide, you get them all added, you wash everything off, you add the next nucleotide, you wash everything off, and then you just look and see. In this particular case, we end up down at the bottom with one sequence which is now blue, red, red, green, green, which is A, C, C, T, T. It's that one specific spot that you see on your microscope slide. And so the key to this is really good optics, actually, to figure out what's going on with these systems. So this is a slide which I pulled up because I thought I did a really crummy job of explaining it the first time around. Basically what we're looking at here is a way of trying to figure out where one particular protein is being expressed. And the way that that's done, you can look at it in two slightly different ways. Probably the easiest way to think about that is down here. This is a way that's an odor receptor that you're looking at in C. elegans. This is basically the nose of C. elegans down here. And then these are that nasal cavity that you have in C. elegans up here. If you just look at a protein fusion to some kind of reporter gene, and the reporter gene in this case is the green fluorescent protein, which is very widely used in cell biology, you can see here that these guys are localized just in the nose. And so you have a reporter construct where you have your favorite protein literally hooked up to the green fluorescent protein, and then you see where your protein is because it's dragging the green fluorescent protein with it. That's one way of looking at it. We talked a little bit about the promoter fusion constructs, and promoter fusion constructs are just the promoter with your reporter gene right next to it. Now here, you're not necessarily going to get where that protein is. You're just seeing where that protein is actually being made. And so in this case, with the promoter fusion, you see that the GFP is actually being made way back here and needs to end up out here at the nose. And so what this tells you is that there's some part of the protein, which you have here in your protein fusion, that tells it, once it's been made here, to get out here. So you're getting two different kinds of information from this experiment. Either the protein fusion, which tells you exactly where that protein is, or the promoter fusion, which tells you where it's being made. So there are two different things that you can get out of this. Gene mapping, again, I think I did a not terribly good job of explaining this before. SNPs are just single nucleotide polymorphisms, and in this particular example, they're just being used for mapping by association. You have a single nucleotide change at one particular position in the 3 billion base pairs in the genome, and you have a phenotype that goes with it. And so this is just the orange phenotype here, and you can see that the orange phenotype, in this case, is really closely associated with this C residue at this one position. So if you have that C residue at that position, you always are going to end up with that particular gene, again, with that particular phenotype. The SNP may or may not be what's causing that phenotype, but at least it allows association. And there are literally millions of different SNPs in the genome that can be used for genetic mapping experiments. Last thing that I wanted to talk about in terms of our genetics are the knock out or how to make knockout organisms. The classic way to do this in mice, start out with embryonic stem cells. This is why people are so concerned about embryonic stem cells in terms of using them for medical applications. There's all kinds of ethical implications that I'm not going to talk about now. But making embryonic stem cells that have a particular mutation in them, basically you use this very much like we talked about for the vectors. You have an antibiotic resistance, but the problem with most mammalian cells, if you put in a vector, that vector usually gets incorporated in a random position, not through homologous recombination, which is what you want. So when you're making these ES cells, you need to do two kinds of selection. You need to select for your antibiotic resistance, but against this non-specific integration that takes place. So you need to do a positive selection, again, for antibiotic resistance, but that will get both homologous recombination and non-homologous recombination, and then select against these guys that have random insertions. Once you've done that, now you have embryonic stem cells which have the particular mutation in them. This could be a knockout. It could also be a knock-in, some kind of gene change which you put in there. Then you take these embryonic stem cells and put them into a blastocyst. This is actually already on its way to making a new organism. So you end up with a chimeric organism. This then has to be bred with a non-chimeric organism in order to get our ES cells, and particularly these are going to be the organisms. Some of these are going to have germline mutations in them, some of them are not. And then you have to screen these and eventually end up crossing to get your knockout down here. And the basic take-home message here is that it's a really long process that you have to go through 6 to 9 months to make a knockout in a mouse. So you have to think about it. And I just heard from one of my colleagues, costs about $1 a day per mouse to keep them in a facility. And so you can do the math, $1 per day per mouse per 6 months to get your knockout mutant. This is a really expensive proposition to talk about here. So any questions about genetics before we talk about chromatin, which is the last couple of lectures? Okay, so bacterial chromatin compaction. This is same problem you always have with DNA. It's just too long. You have to shrink it down somehow. How do you do that? Well, clearly you've got supercoiling which takes place. You have proteins associated with the DNA, but you also have a number of small molecules that are associated with the DNA, and those are particularly those which are positively charged. And so really 3 different ways that you're getting chromosome compaction in E. coli, and also to some extent true in eukaryotic organisms. Eukaryotes have, particularly the larger eukaryotes like us, have way more DNA to deal with, so they've got lots of extra problems to deal with. They also have to have these metaphase chromosomes, which have to be nicely separated. So we need lots and lots of compaction to take place. So we need to go from our double-stranded DNA to nucleosomal DNA, which are really beads on string, 30 nanometer fibers, these loops of 30 nanometer fibers that really lead to the 300 nanometer fibers. And this higher order loops of 30 nanometer fibers, this is what's mostly present in interphase nucleus. So it's 30 nanometer fibers, some of those wrapped up into the 300 nanometer fibers. Beads on a string, this is really what's happening in terms of the actively transcribing, actively replicating DNA. And that's because you have to have access of all these other proteins to that particular DNA. And so this seems to be the way that we're getting compaction, but still accessibility, and that's this ability to go between open and active DNA to closed and inactive DNA. What's in that DNA? Lots of different things, but probably one of the most important of them is to understand what's a gene and what's not a gene. And I just wanted to point out here again that you can just by looking at protein, or for that matter, sequence, nucleotide sequence similarity between different organisms, and if you find a region which is well conserved between these two different organisms, those sequences are likely to be expressed in under selection. On the other hand, something like an intron is not being expressed. It's not under selection. Of course, we've got those other sequences that are also conserved but not being expressed into protein. Those are probably your small RNAs or coding RNA sequences. I mentioned genome synteny before, and again, just terminology-wise, synteny refers to genes that are in the same order relative to each other in multiple different organisms. And so you can see that here nicely for mice and humans. How do you get gene synteny, and how do you get multiple different genes, particularly similar genes to each other? This is the gene family formation. Gene family formation, people predicted, was due to gene duplication. After you have duplicated genes, one of those genes can diverge through mutation. The other gene has to be conserved for that original function, because presumably you still need that particular function. One of the immediate predictions that you have here is that you'll end up with some genes that are non-functional. And so the presence of pseudogenes in these gene families is really good support for this model, which was just a model, that it's gene duplication followed by divergence, which leads to getting much more function and all of these gene families. In our genomes, most of our genome is transposons. There are 3 different kinds of these transposons or jumping genes. Either we've got the DNA-only transposons, which are very common in bacteria and really useful tools in terms of playing around with DNA because they jump around. The retroviral-like retrotransposons, and these have these really interesting long terminal repeats at either end that, again, we'll talk about a lot more in virology next term. But these really look like viruses that have hopped into whatever genome they're in. And if you think about the human genome, it's 8% of our genome which looks like these particular viruses compared to the 1.5% which is protein coding. Then the most common of the retrotransposons that we have in our genome are these non-retroviral retrotransposons. Whether they're related to these viral ones or not is not clear. They have a reverse transcriptase gene because they obviously have gone through an RNA intermediate. But instead of an integrase, which I think I didn't talk about very well, integrase is sort of the retroviral equivalent of the transposase, functions in a very similar way. These guys just have endonucleases, so they just cut in a particular piece of DNA, and then replication happens from that particular point, as opposed to the classic kind of transposon where you grab a piece of DNA and pop it into the genome. So you're having replication that's happening with these individual RNAs that are jumping around there. We talked a little bit about how you can detect these transposons in genomes. The main way is looking at these direct repeat sequences that you have on either side of your transposon. This is outside of the transposon. It's a sequence which came from the original chromosomal DNA that transposon jumped into. It's true for the DNA transposons, and it's true for the LTR retrotransposons. It's actually not true for the non-LTR retrotransposons, so you don't have these direct repeat sequences at either end. This DNA transposons have inverted repeat sequences, so same sequence on opposite strands. And the reason that they have this sequence is that's what that transposase is actually binding to. It's a dimeric or tetrameric transposase, binds to each of those ends, pulls them together, and so that's the piece which actually ends up getting moved and put into the genome somewhere else. The LTR retrotransposons, or the virus-like retrotransposons, have these long terminal repeat sequences, and that has to do with how these are actually getting replicated. They've got a tRNA which serves as a primer. That tRNA is going to copy one end of the genome, and that end of the genome has to recombine with the opposite end. How that happens is not critical, but you do end up with these long terminal repeats, which is what LTR stands for, that is a really diagnostic part of looking at these particular transposons. Another thing about these LTRs, they're extremely good promoters. And so they're great at expressing whatever genes are right next to them. And this is usually a viral genome, if you think about this dark green here. But if you look at this sequence right here, it's identical to this sequence, directly repeated. It's also a promoter, and that promoter can easily be expressing genes that are right next to it, and often expressing genes right next to it that probably shouldn't be being expressed in that high amount. So the activity of one of these LTR retrotransposons jumping into a genome next to a gene which is otherwise not expressed could be a big problem. And it turns out that was one of the first problems people had with gene therapy, because they actually ended up expressing genes they shouldn't have due to the insertion of some of these LTR retrotransposons. Talk about the mechanism here. And again, I just copied these slides directly. This is the mechanism for the non-LTR retrotransposons. And what we're looking at here, so I mentioned that you didn't get the direct repeats. That's true in some cases. Some cases that actually you do get these direct repeats. But it's really all about the endonuclease activity. So not the integrase, which is bound to the ends of your genome, or the transposase is bound to the end of your transposon. Here it's an endonuclease which cuts. If that endonuclease cuts in an offset form, you are going to end up with direct repeats here. But the really important thing here is this has to have a stretch of T residues in it, because those need to base pair with the poly A, which is at the end of the RNA which is being made by these retrotransposons. So those always have poly A tails. Those poly A tails have to bind to T residues where that endonuclease is actually cutting. So these are not quite as random in terms of their integration sites as the LTR retrotransposons and the DNA-only retrotransposons. Once you have that, it's a reverse transcriptase, makes this copy, and then this gets reintegrated into the genome. We talked a little bit about how you can use these transposons as tools. Again, mostly people are using these DNA transposons as tools because they're easiest to work with. They also are relatively nonspecific in terms of where they bind. But you can put all kinds of extra things in between the inverted repeat sequences that you have at the end of your genome and get drug resistance or some kind of reporter gene, or a lot like you have with those LTR retrotransposons, you can put a promoter into one of these transposons and then have that transposon jump into your genome somewhere and just look for genes that get overexpressed. That will be giving you a phenotype, could just be looking at all the expression of any individual genes, and that will give you, since you know where this transposon is, because you can sequence from this known sequence, which is right here, that'll tell you what kinds of genes you get there. This is very often used in genetic screens to try and just figure out what each gene in any particular organism is doing at any one particular time. So last lecture we talked about eukaryotic chromatin. I'm going to go over this really quite quickly. The important things here are, again, what I talked about when I started talking about chromatin. It's about the open structure. We have nucleosomes associated with your DNA and it beads on a string, or a much more compacted case where you're not going to be able to get transcription, replication, etc. proteins into that particular piece of DNA. So this is our active form of DNA, relatively small amounts in the cell. This is the DNA which can be relatively easily activated. These guys go back and forth pretty easily. As soon as this starts to get compacted anymore, then you start to have problems. Each of these beads on the string, of course, is a nucleosome with your 8 core histones in the middle here, 4 sets of dimers. They're very highly conserved because they're all binding to DNA. These also get modified. Almost all the modifications is happening on these tails, which hang out, coming out of the nucleosome. Those then both interact with the DNA, and you can see that happening here as they move out, but they also interact with other histones, through, you can see really nicely this tail right here, which can be interacting with another histone when it's getting packed down inside these 30 nanometer filaments. And then you can see these interactions which are happening right here. Somebody asked me right before lecture whether I consider these SMC proteins as scaffolding proteins, and I said no. And the reason for that is that we haven't really talked about any specific scaffolding proteins, and scaffolding proteins are binding specifically to scaffold attachment regions, which are sequences in your DNA. And the SMCs bind in a very non-sequence specific way. Actually, they serve as clips. They're actually clipping around DNA as opposed to binding to the DNA specifically. And so these clips, the condensins, but also you have similar kinds of proteins in E. coli, the MUC-B proteins, will then clip around the DNA and hold some of these loops together, particularly important for mitotic chromosomes where you really have to have that really massive condensation that takes place. The last thing that I wanted to talk about today is how you get from that open form of DNA to the closed form of DNA and vice versa. Usually this has to do with these histone code reader-writer complexes. Complexes, not surprisingly, they're made up of multiple different proteins, and usually those proteins end up being something that binds to a modified histone, and then another part of that protein complex is something which will modify a histone. So once you have one modified histone, that leads to modifications in the same way of the histones all around that first modification. So once you have that initiating step, you now have a positive feedback loop that leads to that same kind of modification that happens on the histones right there. And so if you have an area of the genome which you need to open up and lead to activation of genes, you would have a histone acetylase, which is going to be acetylating your histones. That's going to be causing the histones to open up. You'll have a histone code reader that will bind to that acetylated histone and also have a histone acetylase associated with it that will lead to that whole region being opened up, because these genes are pretty big. They're a lot bigger than the 140 nucleotides that you have wrapped around one particular nucleosome. Or in the opposite case, if you have a region which you want to be shutting down, now you have a histone methyltransferase, which will put methyl groups onto your histones, and then the proteins that will recognize that and lead to more methylation of those particular histones. So that's all the stuff that's going to be on the midterm. Feel free to email me, come to my office hours today, although I'll be a little bit late because I have to go to the bike shop. And good luck on Monday.